Good afternoon, everyone. Today, we are going to delve into reviewing past A-level examination papers. This session is designed to help you understand how to approach this paper effectively and ultimately enhance your exam performance. We will explore the structure of the papers, common question types, and strategies for analyzing and reviewing them. Without further ado, let's get started. Which row is correct for a typical plant cell? The correct row for a typical plant cell is B. Plant cells have cellulose in their cell wall, a diameter of approximately 5 to 40 micrometers, and contain both 70S ribosomes in the chloroplast and 80S chromosome ribosomes in the cytoplasm. An electron micrograph of a cell shows large quantity of rough endoplasmic reticulum and many Golgi bodies. What type of cells is being viewed? The cells being viewed is most likely mesophyll. Mesophyll cells found in plant leaves are involved in photosynthesis and typically have a large quantity of rough endoplasmic reticulum, so-called RER, and numerous Golgi bodies due to their high protein synthesis and processing requirements. In contrast, bacteria and gut cells do not generally exhibit such features, and lymphocytes, being immune cells, are less likely to have such extensive RER and Golgi networks. Which cell structures can form vesicle? The correct options is A. Both the cell surface membrane and the endoplasmic reticulum can form vesicle through budding processes, and the Golgi body is also involved in vesicle formation, particularly in packaging and modifying proteins and lipids into vesicles for transport. Which sets of measurements is correct? The diameter of a capillary is typically about 0.7 millimeter and approximately around 700 micrometers, and the diameter of the red blood cells is approximately 7 micrometers. The thickness of the cell's membrane is around 7 nanometers. So the correct answer is A. The size of the measles virus is approximately 150 nanometer. The Mimi virus is approximately 4.5 times larger than the measles virus, with the Pandora virus is approximately 1.5 times larger than the Mimi virus. Which viruses can be seen using both a light microscope with a maximum resolution of 0.25 micrometer and an electron microscope? The answer will be measles virus cannot be seen, Mimi virus can be seen, and Pandora virus can be seen. Which carbohydrate give a brick red color when boiled with Benedict solution? When boiled with Benedict solution, fructose give a brick red color. This is because fructose is a reducing sugar and can react with Benedict solution to produce a reddish precipitate, which indicate the presence of reducing sugars. Which two polysaccharides both have 1,6-glycosidic bond and are branched? The two polysaccharides that both have 1,6-glycosidic bond and are branched are amylopectin and glycogen. Both of these polysaccharides have branching structures due to the presence of 1,6-glycosidic bonds, which differentiate them from other polysaccharides like amylose, cellulose, which are unbranched. Number 8 is a common question which exists in October 2017, which is the questions is more related to sucrose, whereas for March 2019 and May 2019, the questions is more related to hexose. The questions is, which two molecules combine to form a molecule of sucrose? The answer is, to form sucrose, C12H22O11, you need to combine two carbohydrate molecules and remove a water molecule. The breakdown, diagram 1, C7H6O6, diagram 2, C2H9O6, and diagram 3, C7H6O6. So when you combine diagram 1 and 2, adding them give C9H15O12. Removing a water molecule, H2O, from this result give C12H22O11, which is sucrose. So the two diagrams that combine to form sucrose are diagram 1 and diagram 2. The diagram shows 3 hexose sugars. This is also a common question which exists in May 2019. So which row correctly show example of carbohydrate in which these 3 hexose sugars occurs? The answer will be diagram 3 is reflect sucrose, and cellulose will be diagram 2, and starch will be diagram 1. The diagram shows the amino acids in a polypeptide. An enzyme catalyzes the hydrolysis of bond between polar basic amino acids and non-polar amino acid. In these questions is asked about how many peptides change of amino acids will be formed as a result of the hydrolysis of this peptide. To find the number of peptide change formed after hydrolysis, you have to follow 3 steps. The first step is identify hydrolysis point, where the enzyme cuts between polar basic and non-polar acid amino acids. There are 10 polar basics and 13 non-polar amino acids. Second, you have to count the cuts, and each cut between polar basic and non-polar amino acid separate polypeptide into smaller chains. The maximum number of this cut is determined by the smaller group, which is polar basic amino acid, roughly estimated about 10. And then step 3, to calculate the number of change. Each cut creates an additional peptide change. So there are 10 cuts. Estimate 10 plus 1 is equivalent to 11 peptide change. Since 11 is not in the option, so the closest answer will be 8. This type of questions is already exists in October 2020. So the questions that related in this paper is about how many small peptides and single amino acid will be formed by the actions of this enzyme. Lysozyme is an enzyme found in tears and saliva, which statements refer only to the tertiary structures of lysozyme. The statements that refer only to the tertiary structure of lysozyme is the 30th and 52nd amino acid help form the active site. So the tertiary structures of a protein involve the 3-dimensional folding of the polypeptide chain, include the arrangements of amino acids that form the active sites. Some RNA molecules called ribosome can catalyze reactions in a similar way to protein enzymes. Most of these ribosomes have other RNA molecules as their substrates and catalyze reactions that break specific sugar phosphate bonds in the substrate molecules. And which statement about these ribosomes are correct? The correct statements is number 2 and number 3. Let's look into number 2. The active sites of ribosomes is made up of a specific sequence of nucleotides. So statement 2 is correct. Statement 3, ribosomes can form because RNA can adopt specific shape, which is secondary and tertiary structures that enable them to function as catalysts. So this statement 3 is also correct. The graph shows the effect of an enzyme on a reaction. Which labels are correct? So in this, refer to this graph, Y is indicate the reactions with the enzyme, and W is the difference in activation's energy. Which statements about non-competitive enzyme inhibitors are correct? The correct statements about non-competitive enzyme inhibitors are all. Means that statement 1, statement 2, and statement 3 are correct. Non-competitive inhibitors can bind to the enzyme regardless of whether the substrate is bound, often at N-allosteric sites, and they inhibit enzyme activity by preventing products formation even if the substrate can still bind to the active sites. The following are all processes that allow movements into cells. Which processes require ATP? Active transport require ATP, which is statement number 2, to move substrate against their concentration gradient. Phagocytes, statement number 1, also require energy, but it involves cellular processes like engulfing large particles, and it's considered a form of endosynthesis rather than a direct use of ATP in the same way active transport does. Statement 3, facilitated diffusion does not require ATP as it relies on the concentration gradient and the existence of transport protein. The fluidity of the cell surface membrane can be changed by a number of factors. As the fluidity of cell surface membrane decreases, which process would be least affected? The answer will be osmosis. Osmosis involves the movement of water through the selectively permeable membrane, which is less dependent on the membrane fluidity compared to the processes like active transport, diffusion, and endosynthesis. The diagram shows the cell cycle, during which phase do chromosomes condense and become visible. Chromosomes condense and become visible during prophase, which is represented by 25% of the cycle. A reflect typically anaphase or telophase in the cell cycles, where chromosomes are either moving to opposite poles or starting to decondense. B represent interphase, when chromosomes are in their more relaxed chromatin state and not visible as distinct structures. D represent metaphase, where chromosomes align at the cell's equatorial plane but are already condensed and visible from prophase. So the correct answer will be during prophase, chromosomes are condensed and visible, which is C. Colchicine is a chemical that stops chromatid from separating during mitosis. A cell is treated with colchicine. Which stage of mitosis will these cells reach but not complete? The answer will be metaphase. Colchicines inhibit the formations of the mitotic spindle, preventing the separations of chromatid during anaphase. Therefore, chromosomes can align at the metaphase plate, but the cells cannot proceed to anaphase or complete mitosis. Question number 18 is already repeated in May 2021. The following statements describe the process of translation. In which order does this process take place? First is mRNA beats to the chromosome, a ribosome, and next, followed by tRNA enters the ribosome carrying a specific amino acid, and then hydrogen bonds form between the anticodon and the codon, and finally we have a peptide bond formed between adjacent amino acids. Which row represent the correct features of the nitrogenous base adenine? This is also a common question. And in paper May 2018, but the questions during at that time is asking about guanine. And the answer for adenine is C. The codons UGU and UGC code for the amino acid cysteine, which can form the sulfate bond in a polypeptide. The codon UGG codes for the amino acid tryptophan, which does not contain a sulfur atom. The codon UGA is a stop signal. The DNA triplet codes for the 10 amino acid in a particular polypeptide is ACA, which a single basic substitution in this triplet code will result in no disulfide bond being formed with the 10 amino acids in the polypeptide. To ensure the 10 amino acid does not form disulfide bond, it should not be cysteine. The original DNA template ACA codes for threonine, which does not form disulfide bond. The following substitution also code for threonine, which is AGG, AGG, ACG, ACT. Since all these substitutions result in threonine, none will form disulfide bond. Therefore, the correct answer is ACC and ACG. A student produced a table of differences between RNA and DNA. The tables contain one mistake. Which row has the mistake? The row with the mistake is C. While it is true that DNA is predominantly found in the nucleus in eukaryotic cells, RNA can also be found in the nucleus as well as in the cytoplasm. The correct distinction is that DNA is primarily located in the nucleus, while RNA is found in both the nucleus and the cytoplasm. The diagram shows transverse sections of part of a plant with some transport tissue labeled 1 until 6. This is also common questions. It already exists in March 2019. Which tissue mainly transport water and which tissue mainly transport sucrose? Tissue that transport water will be 2, and tissue that 2 and 3, and tissue that mainly transport sucrose will be 4 and 5. Transpiration is a consequence of which features of plants? Transpiration is the consequences of the requirement for leaf to exchange gases. And it is primarily occurs due to the need for gas exchange in leaf. As plants take in carbon dioxide for photosynthesis, water vapor exits the leaf through stomata, leading to transpiration. Mass flow is the bulk movement of material from one place to another. How many of the vessels listed carry fluid by mass flow? The answer will be folium sieve tube and xylem vessels. In plant, mass flow occurs in the folium for the transport of nutrients and in the xylem for the movement of water and mineral. Arteries and veins in animals primarily use pressure-driven flow rather than mass flow. The diagram shows a xerophytic leaf in different condition P and Q, which statement describes the difference between the cells in layer X in condition P and Q. The answer will be move negative water potentials in P than Q, then move cells plasmolyzed in P. This is also the common questions exist in May 2021. What would increase the rates of transpiration? To increase the rate of transpiration, you would increase the light intensity. Higher light intensity increases the rate of photosynthesis, which leads to more stomata opening and thus higher transpiration rates. Increasing humidity or decreasing temperature and wind speed would generally reduce the rate of transpiration by decreasing the water vapor gradient between the inside of the leaf and the surrounding air. What happened during ventricular systole in the mammalian heart? During ventricular systole in the mammalian heart, aortic pressure increases. Ventricular systole involves the contractions of the ventricles, which increases the pressure within them. And this increased pressure forces blood out of the ventricles and into the aorta and pulmonary artery, leading to an increase in aortic pressure. During this phase, the atrioventricular valves, which separate the atria and ventricles, close, and the semilunar valves, which separate the ventricles from the aorta and pulmonary artery, which is open to allow blood to be ejected. Which substances would displace oxygen from oxyhemoglobin? The substances that could directly displace oxygen from oxyhemoglobins are carbon monoxide and hydrogen ion. The carbon monoxide can bind strongly to hemoglobin, displacing oxygen. Hydrogen ion reduces hemoglobin affinity for oxygen, promoting oxygen release. Carbon dioxide and hydrogen carbonate ions influence oxygen binding indirectly through the Bohr effect, but do not directly displace oxygen. The graph shows the affinity of hemoglobin for oxygen at two different concentrations of carbon dioxide. Which point on the graph show the percentage saturations of hemoglobin entering the pulmonary artery? In the pulmonary artery, the blood has a low partial pressure of oxygen and high carbon dioxide, leading to low hemoglobin saturations with oxygen. So, the point on the graph that best represents this situation is at B. The diagram shows some of the reactions of carbon dioxide when it enters the blood from cells in a metabolically active tissue, which reactions is catalyzed by the enzyme carbonic anhydrase. This is also a repeated question and occurs in October 2021. The answer will be C. How many phospholipid bilayers does oxygen pass through in diffusing from an alveolar air space to form oxyhemoglobin in the red blood cells in a mammalian lungs? Oxygen diffusing from an alveolar air space to form oxyhemoglobin in the red blood cells passes through 5 phases. So the path as stated below, first path is alveolar air space across the alveolar epithelium. Path 2, alveolar epitheliums, 1 phospholipid bilayer. Path 3, interstitial space between alveolar epithelium and capillary endothelium. And path 4, capillary endothelium, 1 phospholipid bilayer. And path 5, red blood cell membrane, which is 1 phospholipid bilayer. And each layer consists of a phospholipid bilayer, which is come into a total of 5 bilayer. Which statements about the human gas exchange systems are correct? The answer will be statement 3 and statement 4. Alveolar secret surface tension, which reduces surface tension in the lung. The trachea and bronchi are supported by circles of cartilage. Statement 1 is incorrect because the absence of cartilage in small bronchi make them less stable and prone to collapse rather than allowing expansion. Statement 2 is also incorrect because the wall of the alveoli is made of a single layer of squamous epithelium, not cuboidal. A disease damages alveolar walls, causing the alveoli to burst. What effect does this have on the gas exchange system? If a disease damages the alveolar walls, causing the alveoli to burst, the effect on the gas exchange systems would be surface area for gas exchange decreases, and the maximum volume of the air breathed out is also decreases. Damage or burst alveoli reduces the surface area available for gas exchange, impairing oxygen and carbon dioxide exchange. This damage can also affect the lung elasticity and capacity, which lead to a decreased maximum volume of air that can be breathed out. What are the short-term effects on nicotine on the cardiovascular system? The short-term effect of nicotine on the cardiovascular systems are on constriction of the small arteries, increase in heart rate, and increase in the blood pressure. And this effect contribute to the immediate impact of nicotine on cardiovascular health. What is the correct for tuberculosis? The correct answer is airborne transmission. Tuberculosis is caused by a bacterium, Mycobacterium tuberculosis, and is transmitted through the air when an infected person coughs or sneezes. It is not caused by a virus and does not typically present with a rash. Which factors contribute to outbreak of measles after natural disaster? The answer will be statement 2 and statement 3. A lack of effective vaccination coverage in the population before the disaster and people living in overcrowded accommodation both contribute to the spread of measles. Contamination of drinking water with untreated sewage is more relevant to waterborne diseases rather than measles. Which of these statements explain why some vaccines can be taken by mouth, but tuberculosis vaccine has to be injected? Statement 2. The TB antigen necessary to produce an immune response are protein which would be digested in the stomach and small intestine. The TB vaccines contain protein that would be digested if taken orally. Therefore, the vaccine must be injected to bypass the digestive system and effectively stimulate an immune response. Which method of gaining immune can be described as natural active immunity? The correct answer is inhaling the chickenpox virus. Natural active immunity occurs when you are exposed to a pathogen like the chickenpox virus, and your immune system responds by producing its own antibody and memory cells. Which statement about endocytosis are correct? The correct statements is 1, 3, and 4. Reason being for statement 1 is endocytosis include processes like phagocytes. Statement 3, endocytosis involve taking materials into the cells. And statement 4, vesicles form within the cytoplasm during endocytosis. And meanwhile, statement 2 is found to be incorrect because endocytosis is an active process, not passive. And the statement referred to vaccination program for each of the disease: cholera, measles, smallpox, and tuberculosis. Which statement is correct for TB? The correct answer is the vaccine is not effective against some strain of the pathogen, because the TB vaccines may have variable effectiveness against different strains of a tuberculosis bacteria. That's all for today's sessions. Hope that you are doing well in your studies and upcoming examinations. And please don't forget to subscribe to Jom Stabila YouTube channel and on your notifications so that you can get the latest updates. And thank you very much for your support and cooperations.